TissueLyser Machine is a vibration mall, disrupt biological samples in parallel, by using microtube rack and caps with two adapters, and each adapter comprise top and bottom plate, for holding microtube rack, as mentioned in next slides. TissueLyser Machine, initiate by place target sample in 2 ml microcentrifuge tube, contains 2 grinding stainless steel balls. After that, add appropriate volume of lysis buffer to each tube, and place microtubes in TissueLyser adapter, and operate at 2 to 4 minutes, with 20 to 30 Hz, until tissue debris visible, as mentioned in next slides. During work with TissueLyser Machine, don't operate TissueLyser during transport, and don't autoclave any parts of tissue layer, and clean TissueLyser by moist cloth, and clean adapters with 70% ethanol. So, TissueLyser Machine, consider fast and effective disruption multiple samples at once, by provide high-speed shaking with oscillation frequencies, to break down complex tissues into uniform, to achieve highly reproducible purification. On the other hand, TissueLyser Machine, consider labor and costly method, and require experts during operation to avoid any damage. Therefore, TissueLyser Machine, use in genetic studies, and molecular biology studies, and life science studies, and biochemistry studies, and pharmaceutical studies. On the other side, pestle and mortar is traditional method, crush and grind substances into fine powder, to reduce particle size of solid substance, by pound, press, and circular motion, to improve solubility and reaction rates, as mentioned in next slide. There are many types of pestle and mortar, which are stone, and wood, and glass, and metal, and ceramic, and porcelain. Pestle and mortar steps: initiate by rinse mortar and pestle with warm water, and fill 1/3 mortar with substances to avoid spillage, and use one hand to hold mortar steady on flat surface, and crush substances between mortar and pestle, and use circular motion to achieve desired consistency, as mentioned in next slide. And clean mortar and pestle immediately after finish by warm water and soft brush, and dry mortar and pestle thoroughly with paper tissue, and store mortar and pestle in dry place, as mentioned in next slide. Therefore, pestle and mortar, consider relatively inexpensive method, in generate very small particles. On the other hand, pestle and mortar, lead contaminant issues from dust during work. Finally, pestle and mortar, use in genetic studies, and molecular biology studies, and life science studies, and biochemistry studies, and pharmaceutical studies.